Hey there, and welcome. In this explainer, we are diving deep into one of the most important experiments for your GCSE biology exam. Yep, it's required practical number 6, all about the rate of photosynthesis. Trust me, if you can nail this practical, you're well on your way to nailing that paper. So, how on earth can a little piece of pondweed help you pass your GCSEs? Sounds a bit weird, right? Well, it's the absolute star of this experiment, and understanding how it works and what it shows is a surefire way to hoover up a ton of marks. Okay, let's get into it.
So, here's our plan. First up, we're going to cover the core science behind everything. Then, we'll walk through the practical itself step by step. After that, we'll look at what to do with your results, and to finish, we'll focus on those all-important exam skills you need to really get the best grade you can.
Right then, Section 1, the core principle. Before we even think about touching a beaker or a stopwatch, we have to be absolutely crystal clear on the science that this whole investigation is built on. So let's start with the big one, photosynthesis. It's that endothermic reaction plants use to make their own food. Now that word endothermic, that's a key exam term. Don't let it throw you. It just means the reaction sucks in energy from its surroundings. And it's all summed up in that famous word equation you see right here. Carbon dioxide plus water, with some light energy, gives you glucose and oxygen. Now, here's where it gets really interesting. The speed or the rate of photosynthesis isn't always flat out. It can be held back. And the one thing that's holding it back is called a limiting factor. Think of it like a massive traffic jam on a motorway. It doesn't matter that your car could go at 70 miles an hour. That bottleneck sets the speed for everyone. And for photosynthesis, there are 4 main suspects that can act as that bottleneck: light intensity, CO2 concentration, temperature, and how much chlorophyll the plant has. In this practical, though, we're going to put just one of these under the microscope: light intensity. Which brings us nicely to Section 2, the Pondweed practical. This is your how-to guide, your step-by-step for doing the experiment and getting some really solid, reliable results in the lab. Here's the standard kit you're going to need. You've got your piece of pondweed, a beaker of water, a lamp of some kind, a ruler to measure the distance, a stopwatch for timing, and this chemical here, sodium hydrogen carbonate solution. It might seem a bit random, but it's actually super important. And here's why. This is a huge exam tip, so listen up. You add the sodium hydrogen carbonate to the water because it provides a steady, constant supply of carbon dioxide for the pondweed. See, we want to investigate how light affects the rate, so we have to make sure that a lack of CO2 doesn't become the limiting factor instead and completely mess up our result. The method itself is pretty straightforward. First, you set up your lamp at a set distance, let's say 10 centimeters. You then let the pondweed just sit for a few minutes to get used to the conditions. Then you start the clock and count the tiny bubbles of oxygen it produces in 1 minute. And this is crucial. You do that 2 more times and work out the average. That makes your results way more reliable. After that, you just move the lamp further away to 20 centimeters, 30 centimeters, and so on, and repeat the whole process. Okay, on to Section 3. You've done the experiment, you've been counting bubbles like a champion. Now what? Well, this part is all about turning those raw numbers into something meaningful and eventually into a graph that's going to score you top marks. Your results table should end up looking something like this. See how there are columns for each trial, and then a final one for the mean or the average? Doing repeats and calculating a mean is just so important for making sure your results are reliable, and it also helps you spot any weird, anomalous results that just don't fit the pattern. The pattern you should see is usually pretty clear. As the lamp gets further and further away, the light that actually reaches the pondweed gets less and less intense, and this shows the relationship perfectly. Less light energy means a lower rate of photosynthesis, which we see as fewer oxygen bubbles being produced every minute. Simple as that. Now, when it comes to plotting this data on a graph, presentation is everything. On the left, you've got a perfect example. The axes are clearly labeled with units, the scale uses up most of the paper, the points are plotted accurately, and there's a lovely smooth curve of best fit. But on the right, it's a disaster. No labels, a cramped scale, and worst of all, they've joined the dots. Never, ever join the dots. This stuff is where easy marks are won or lost. Okay, higher tier students, listen up. This next bit is just for you. There's a specific mathematical rule you need to know called the inverse square law. See, light intensity doesn't just get weaker with distance, it follows this specific pattern. Basically, it means if you double the distance, the light intensity drops to 1/4 of what it was. And if you triple the distance, it drops to just 1/9. It's a key detail that'll help you get those top grades.
And that brings us to our final section, which you could argue is the most important one of all, acing the exam. Let's break down exactly what the examiners are looking for when they ask about this practical.
Right, you absolutely must be able to identify the variables. The independent variable is the one thing you are deliberately changing. Think I for independent, I for eye change. In this case, it's the light intensity, which we change by moving the lamp. The dependent variable is what you measure to see the effect. It depends on what you changed. And here, that's the rate of photosynthesis, which we measure by counting the bubbles. Now, to make sure this is a fair test, every single other thing that could possibly affect the results has to be kept the same. These are your control variables. Things like the temperature of the water, the concentration of CO2, using the exact same piece of pondweed for all your readings, and making sure you count the bubbles for the same amount of time every single time. All of them are crucial. Examiners love asking you to evaluate the experiment, to pick holes in it. So what could go wrong? Well, the bubbles might come out so fast you can't count them accurately, or maybe the bubbles aren't all the same size. A big one is that a hot lamp could heat up the water, which would change the temperature, and that would mean you're changing two variables at once, making your test totally invalid.
But for every potential error, there's an improvement. To make it more accurate, instead of counting bubbles, you could collect the oxygen gas in a measuring cylinder that's upside down in the water. Measuring the volume is way better. To improve the validity, you could use an LED lamp that doesn't get hot, keeping the temperature constant. And to improve reliability, that's an easy one. Just take more repeat readings at each distance.
So the crucial point is this. It's the absolute heart of all good science, and it's what this practical is all about. For your results to be valid, you have to change one thing, your independent variable, measure the effect on your dependent variable, and keep every other possible control variable exactly the same. And that's it. That's your complete guide to the photosynthesis practical. But I want to leave you with one final thought. We've seen how light can be the single factor that limits a plant's growth. So my question to you is, what else, maybe in nature or even in our own lives, is held back by just one single limiting factor? Something to think about. Thanks for tuning in, and good luck with your revision.